Assalamualaikum everybody, I hope you all are doing well. We are diving into a new and most awaited video series, the Mycology. This is the very first video in Mycology series and it is introduction. But before getting into the video, I'd like to tell you guys that these videos are meant for educational purposes. Things and treatments may change with time. If I get wrong or miss anything, your input is always welcome in the comment section. So grab your snacks and let's get started. The word mycology has got two different words incorporated in it. First one is myco, that means fungi, and the second word is logy, that means to study. So mycology is the study of fungi. You can see a mushroom right there. The reason is mushroom is also a fungus. Fungi are responsible for causing mycosis in human beings. There are three major kinds of mycosis: cutaneous and subcutaneous, then we've got systemic, and the last one is opportunistic. Fungi form spores after sexual or asexual reproduction. If the spores are formed after sexual reproduction, they are termed as sexual spores. And if spores are formed after asexual reproduction, they are termed as asexual spores or conidia. Fungi are eukaryotic organisms. The word eukaryotic also has got two words in it. The word eu means true, and karyotic is derived from karyon, which means nucleus. So eukaryotic means the true nucleus. So fungi are those organisms that have the true nuclei in them. Most fungi are obligate aerobes. Some are facultative anaerobes, but none are obligate anaerobes. All fungi require a preformed organic source of carbon, hence their frequent association with decaying matter. Modes of reproduction. Fungi reproduce by two modes. One is sexual and the second one is asexual. Let's start by talking about sexual reproduction. Some fungi reproduce sexually by mating and forming sexual spores. Sexual spores can be zygospores, ascospores, and basidiospores. We'll talk about them in detail in just a moment. And fungi that do not form sexual spores are termed imperfect and are classified as fungi imperfecti. Asexual reproduction, it's a process by which a single parent cell produces genetically identical daughter cells. These daughter cells develop into new organisms. For example, yeasts. They grow as single cells that reproduce by asexual budding. Here, I would like to take a moment to thank Picmonic for sponsoring today's video. Picmonic is an audio-visual learning platform that is tailored to assess the needs of medical students, physician assistants, doctors, nurses, and more. They provide valuable resources like video lectures,
Crypt keeper spore is a protozoan pathogen shown as the propeller,
visual mnemonics, quizzes, study scheduler, and much more. We'll be looking at the fungi basics from Picmonic in today's video, so stay tuned for that. And also, if you guys are interested in signing up on Picmonic, I've got you a discount code medzukhruf that you can use at the checkout, and the discount link given in the description below that will give you 20% off on your purchase. So hurry up and sign up. Now let's have a look at the classification of fungi. Fungi are classified based on sexual spores formation and also on cell and filament formation. If we look at the classification based on sexual spores formation, we've got zygospores, ascospores, basidiospores, and fungi imperfecti. I've got a really cool mnemonic for that, and that's FABS from fabulous. And now let's look at the classification based on cell and filament formation. I've got a really cool mnemonic for that, and that's YUM. Y is for yeasts, and M is for molds. Now let's have a look at zygospores. Zygospores are single large spores with thick walls. Ascospores. Ascospores are formed in a sac called ascus. Basidiospores. These are formed externally on the tip of pedestal called basidium. Yeasts. Yeasts grow as single cells that reproduce by asexual budding. Molds. Molds grow as long filaments called hyphae and form a mass that is termed as mycelium. The filaments are called hyphae and these are functional unit of fungi. Lecture outline. We are done with the introduction and classification. Now we'll be having a Picmonic review of fungi basics. Then we'll look at the morphology of fungi, habitat, transmission, and at the end, as usual, we'll review the lecture. So this is the fungi basics visual mnemonic from Picmonic. And we start by representing fungi as the fungi. The functional units of fungi are hyphae, shown as the high five with hyphae, which are filamentous extending structures. A mass of hyphae are called mycelium, the mouse lion, and are the fungal equivalent to a bacterial colony. Septa, the scepta, are internal wall segments and dividers which separate hyphae into individual cells. Fungi have cell walls composed of chitin, depicted as a chicken, which maintain the integrity of intracellular environment while protecting the cell from damage. These organisms are saprophytic, the sap, because they live off dead or decomposing matter. They release digestive enzymes, illustrated as the enzyme fish, and later absorb the digested matter from their food source. Fungi can reproduce in two different ways. Sexual reproduction can occur the sex sides high-fiving, and this takes place during unfavorable environmental conditions, leading to fusion of two hyphae of compatible fungi. In sexual and asexual reproduction, haploid spores, the hat-plate spores, are dispersed and grow into new fungi. One type of fungi, yeast, undergoes budding, shown as the yeast budding fungi. Budding is a type of asexual reproduction in which two identical daughter cells bud from the original yeast cell. So, let's summarize the basics of fungi. The functional units of this organism are called hyphae, which are filamentous extending structures. A mass of these hyphae are called mycelium. Fungi are composed of chitin, and these organisms are saprophytic, meaning their food source is dead or decaying organisms. They release digestive enzymes to break down matter, which is later absorbed by the fungi. Remember that septa divide hyphae into individual cells. Fungi can participate in asexual as well as sexual reproduction. Haploid spores are released, which grow into new fungi. Yeast, a type of fungus, undergoes budding to create daughter cells, and this is an example of asexual reproduction. And if you guys are interested in signing up on Picmonic, I've got you a discount code medzukhruf that you can use at the checkout, and discount link given in the description below. That will give you 20% off on your purchase. So what are you waiting for? Go, sign up, and have fun learning. Morphology. Shape. Fungi are tubular, elongated, and thread-like filamentous structures. They are arranged as single cells, for example yeasts, or long filaments, for example molds. Mushrooms are also fungi, and they've got umbrella shape. Fungi vary in diameter from 2 to 10 micrometers. They vary in color from yellow to red to blue, and many others. This is how a fungi looks like under the microscope. This is its long filament. There we go. Structure. Fungi has got four-wing structures: cell wall, cell membrane, hyphae, septa, mycelium, sexual spores, and asexual spores are termed as conidia. The fungal cell wall consists primarily of chitin, not peptidoglycan as in bacteria. Thus, fungi are insensitive to certain antibiotics, such as penicillins and cephalosporins, that inhibit peptidoglycan synthesis. Chitin is a polysaccharide composed of long chains of N-acetylglucosamine. The fungal cell wall contains other polysaccharides as well, the most important of which is beta-glucan, a long polymer of D-glucose. The medical importance of beta-glucan is that it is the site of action of antifungal drug, caspofungin or caspofungin. Next one in the list is cell membrane. The fungal cell membrane contains ergosterol, in contrast to human cell membrane which contains cholesterol. The selective action of amphotericin B and azole drugs such as fluconazole and ketoconazole on fungi is based on this difference in membrane sterols. The third thing in the list is hyphae. These are the functional units of fungi. Singular is hypha and plural is hyphae. These are long filaments. Some hyphae form transverse walls, the septate hyphae, whereas others do not, which are non-septate hyphae. Non-septate hyphae are multinucleated, choanocytic. The growth of hyphae occurs by extension of the tip of hypha, not by cell division all along the filament. Septa. These are internal wall segments or dividers. These separate hyphae into individual cells. Mycelium. Hyphae form mycelium. The mass of hyphae is termed as mycelium. Sexual spores. Some fungi reproduce sexually by mating and forming sexual spores. And we've already discussed the sexual spores, zygospores, ascospores, basidiospores, and those fungi who do not form sexual spores are fungi imperfecti. Now let's talk about conidia. Most fungi propagate asexually by forming conidia, the asexual spores, from the sides or ends of specialized structures. The shape, color, and arrangement of conidia aid or help in the identification of fungi. Some important conidia are arthrospores, chlamydospores, blastospores that include pseudohyphae, and sporangiospores. Arthrospores, which arise by fragmentation of the ends of hyphae and are the mode of transmission of Coccidioides immitis. Chlamydospores, which are rounded, thick-walled, and coit-resistant. The terminal chlamydospores of Candida albicans help in its identification. The third one, blastospores, which are formed by the budding process, by which yeasts reproduce asexually. Some yeast, for example Candida albicans, can form multiple buds that do not detach, thus producing sausage-like chains called pseudohyphae, which can be used for identification. And the last one, sporangiospores, which are formed within a sac, that is called sporangium. Sporangiospores are formed by molds within a sac, and sac is present on a stalk. And which molds form that? Rhizopus and mucor form. Habitat. The natural habitat of most fungi is the environment. An important exception is the Candida albicans, which is a part of human normal flora. We can say that some fungi are found in environment while others are found in human body. Several fungi are thermally dimorphic. Thermally is related to temperature. Dimorphic, di is for two and morph is for shape. These fungi form different structures at different temperatures. They exist as molds in environment at ambient temperature and as yeasts or other structures such as spherules or coccidioides in human tissues at body temperature. Fungi are transmitted via direct contact with spores, like if a person inhales these fungal spores, or if these spores get into direct contact with a wound on a skin or through mouth, and then from mouth they get into GIT, or these spores get directly into the human body through vagina. Alright guys, let's have a quick recap. The word mycology has got two words in it. First is myco, that means fungi, and second is logy, that means to study. So mycology is the study of fungi. Fungi cause mycosis. There are three kinds of mycosis: cutaneous and subcutaneous, opportunistic, and systemic. Fungi form sexual and asexual spores. Sexual spores are formed after sexual reproduction, and asexual spores are formed after asexual reproduction, and asexual spores are also called as conidia. Fungi are eukaryotic organisms. Fungi reproduce by sexual and asexual reproduction. And that's it for today's video. I hope you enjoyed it. If you've got any suggestions, feel free to leave them below in the comments. And if you want to connect with me on my social media, I've got my Instagram and Twitter, both with the handle medzukhruf. And I'll catch you in the next video. Assalamualaikum.